Hello friends, welcome to my tutorial channel once again. Again, this is the question and answer session for genetics. The figure that you can see over here, question is, what is the figure given? The figure is Y-shaped structure, that is a replication fork. What is the site A? This is the site A where the replication begins. So site A is Ori site. Which enzyme acts over here? The enzyme that opens the helix acts over here. The enzyme's name is helicase. Now what are B? These are B. What are they? They are small fragments of newly formed DNA known as Okazaki fragments. Name the strands X and Y. Here X, here Y. X is the strand where continuous DNA is synthesized and Y is the strand where discontinuous DNA are synthesized. That means Okazaki fragments are present. So X is leading strand, Y is lagging strand DNA. This is leading strand DNA, this is lagging strand DNA. Now what is lagging strand made up of? Lagging strands are made up of nothing but Okazaki fragments. Next question is, who gave the cloverleaf model of tRNA? Cloverleaf model of tRNA was given by Hawley et al. Hawley and his co-workers. What is present at its 3' end and at 5' end? 3' and 5'. At the 3' end, guanine residues are present. At the 5' end, CCA bases are present. CCA, cytosine, cytosine, adenine. Name its ribosome binding loop. The name of the ribosome binding loop is T-Psi-C loop. What do you mean by Shine-Dalgarno sequence? It is a ribosomal binding site in prokaryotic mRNA located just before the initiation codon. Initiation codon means AUG. So just before AUG there is a sequence of 8 bases. What is that 8 base? It is a purine rich 5 prime A G G A G G U. This is the sequence. Now what is its significance? It is a ribosomal binding site in prokaryotic mRNA located just before the initiation codon AUG. These are the sequence of bases present in over here. Its significance is to help recruit the ribosome to the messenger RNA. To the mRNA. Fine. Now define operon. What is an operon? Operon is a segment of DNA which regulates the metabolic activities of the cell. Operon consists of promoter, operator, regulator and structural genes. Okay, that you don't have to write, just this is the definition for operon. It is a segment of DNA which regulates the metabolic activities of the cell. Now what are the roles of promoter, operator and regulator genes in an operon? Promoter. It is a site where RNA polymerase binds. RNA polymerase binds to initiate transcription. Operator. Actually, this is the sequence that can bind the expression protein to regulate transcription. It helps to bind the repressor molecule. Regulator. It produces repressor proteins which binds at the operator. Now mention the role of EFTU. EF means elongation factor, TU means thermal unstable, and EFG, this is a translocase factor. Role of EFTU. What does EFTU do? What is its job? It helps to elongate the chain of polypeptide. And EFG is translocase. What does it do? It helps the ribosome to move from codon to codon. On mRNA. Now coming to non-ambiguous nature. Here non-ambiguous means single codon codes for only one amino acid. Okay, there are exceptions. Now about this pedigree. What kind of trait is shown? You can see over here that here it is affected and this generation is not affected. Again this generation is affected, this generation is not affected. If it skips the generation, it means that it is recessive trait. Okay, that means if it is skipping the generation, one is affected, other is not, that means it is recessive. Dominant traits never skip generations. Here male and female both are affected. Here female is affected, here male is affected. So if male and female both are affected, it is autosomal. So the trait is autosomal recessive trait. Give two reasons. So these are the reasons. Reason number one is it skips generations. It skips generations. And number two, both male and females are affected. Next question is describe post-transcriptional processing of RNA in eukaryotes. What is that? It is the changes in primary transcript or hnRNA to mature into functional mRNA. The steps involved are capping with methyl guanosine at the 5' end and addition of polyadenylate residues poly A at the 3' end. This is tailing. Third is splicing of introns, means removal of introns with the help of spliceosome proteins. And fourth is joining of exons. Exons are joined with the help of ligase enzyme. Explain industrial mechanism. Before industrialization, the number of white peppered moth, this concerned with peppered moth. Biston and Betullaria were common before industrialization when the background was white. After industrialization, number of black moth became common, that is Biston carbonaria, which increased a lot due to the environment going black. The black moth got selective advantage to camouflage with dark trees. So this is all about industrial mechanism. Mention the characteristic features of Cro-Magnon man. Brain capacity, cc. Number 2 is elongated nose. Third is well developed chin. Fourth is completely erect posture. Fifth point is highly social, communicative. And number 6 is used to do cave paintings. Give the symptoms and causes of Down syndrome. Down syndrome is known as mongoloid EDS. Number 1 is moon-shaped round head. Moon-shaped head. Number 2, mentally retarded. Number 3, mentally retarded means idiots, dwarfs, dwarfism. Number 4 is protruded tongue. Cause is trisomy of 21st chromosome. Chromosome number 21st. What is the contribution of Sutton and Boveri? They gave chromosomal theory of inheritance. What are pleiotropic genes? Coming to pleiotropism, pleiotropic genes means a single gene that governs multiple phenotypic effects. So this is the end. Thank you for your patience. Please like and share this video and communicate with as many people as possible. And do not forget to press the bell button so that you get proper information. Thank you.